Hello everyone, welcome back to Dr. K. Primer video lecture series presenting by Dr. K. Prem, that's me. Today we'll talk about homopolymer tailing. So homopolymer tailing is a DNA joining method or DNA joining molecule. In addition to homopolymer tailing, you can use linkers and adapters for in vitro ligation of blunted DNA molecules. When you have two different blunted DNA molecules, you can use linkers, adapters, or homopolymer tailing to in vitro ligation of them. So these DNA joining molecules can add cohesive ends on blunted DNA fragments. So in a previous lecture, we talked about the linkers, adapters. In today's lecture, we'll talk about homopolymer tailing. You can see here, this is one DNA molecule, and this is another DNA molecule. So both of them are homopolymerized nucleotides at the 3' end. When you mix these two different DNA populations and they will anneal wherever there is a complementary region and form a recombinant DNA molecule. You can see here, right? A and A is there on one DNA molecule and T is there at the another DNA molecule. When you mix them, they'll find wherever there is a complementary region, and that structure, that annealing allows the formation of hydrogen bonding. That bonding is enough to make a DNA molecule, a recombinant DNA molecule. So in the next slide, we'll see how this process, what is the procedure of homopolymer tailing and how can you use the homopolymer tailing DNA fragments in a molecular cloning. You can see here, this is a blunted DNA fragments with the 3' OH and 5' phosphate, and they don't have any cohesive ends, right? So if you incubate blunted DNA fragments with the terminal deoxynucleotide transferase, that's nothing but TDT, which is a template independent polymerase. This enzyme can add the nucleotides at 3' OH of the substrate without support of the template. So just you have to provide the single strand or double strand DNA and TDT, that's a terminal deoxynucleotide transferase, and add deoxynucleotides. Then those deoxynucleotides are polymerized at a 3' end stepwise. Here you can see terminal deoxynucleotide transferase can catalyze the stepwise addition of deoxynucleotides at 3' OH of single strand or double strand DNAs. So we are using terminal deoxynucleotide transferase for homopolymer tailing or tracks of the blunted DNA fragments. So if you add 4 nucleotides, they'll be added at 3' OH. If you provide only one nucleotide of your interest, that also can be polymerized at 3' OH and forms the homopolymers. So here we are incubating the blunted DNA fragments which are having the 3' OH. That's a most important. And terminal deoxynucleotide transferase and its cofactor, magnesium or cobalt, you can use it. Then your desired nucleotide. Here we are using the deoxyguanosine triphosphate, DGTP. Then if you incubate the reaction for a while, and then homopolymers you can see at the 3' OH of the blended DNA fragments. So terminal deoxynucleotide transferase can add this DGTP at 3' OH of the substrate. You can see here. You see, this is a homopolymer tailing at 3' region of blended DNA fragments, right? And usually the DNA fragment with the poly G at 3' end, usually the polymer, which is a homopolymer at 3' end, is having a length of 10 to 40 bases. So if you incubate with a higher concentration of enzyme, you can get the bigger, longer also. But for homopolymer tailing, which is we are doing for blended DNA fragments, is ideally that length will be 10 to 40 bases. That's enough for the molecular cloning. Hence, so once you are done with the homopolymering of blended DNA fragments, then we have to take the linearized vector. Whatever vector you have chosen, that has to be digested and form a linear with the 3' OH and 5' phosphate. So again, similar like the blended DNA fragments, the linearized vector also incubated with the terminal deoxynucleotide transferase and complementary nucleotides. The insert is homopolymerized with the polyguanine, right? Here, what is the complement for guanine? Cytosine. So here, the linearized vector is incubated with the DCTP and terminal deoxynucleotide transferase and allowed the reaction to continue. Then you can see the vector with the DC tails. You see? Vector with the DC tails at 3' OH region. So I'm telling you all, terminal deoxynucleotide transferase has an ability to add the nucleotides at 3' end of substrate, 3' OH of the substrates. So that's a principle we are using to create a homopolymer tracks or tails at blended DNA fragments. Here, linearized vector incubated with the terminal deoxynucleotide transferase and the DCTP. Once the reaction is completed, you can see the vector with the DC tails at 3' end. Now your insert with the poly G and vector with the poly C are available. If you mix both insert and vector and mix them, then what will happen? They'll search for the complementary region in the DNA, right? Here, the insert is having the poly G and vector is having the poly C at 3' ends. So annealing of complementary homopolymers DC and DG tails of vector and insert. When you mix them, then what will happen? The homopolymer of DC, homopolymer of DG, they will anneal and they form the hydrogen bond between them. So when the homopolymers are having the 20 new bases at 3' end, that's enough to make a intact structure. So once you mix the vector and insert with the homopolymers, they'll anneal and hybridize, and that structure, that intact structure, you can transform into the E. coli. So once you transform into E. coli, then what will happen is there are some discontinuities or gaps or nicks in the that recombinant DNA molecule. So they are filled by the host DNA polymerase, and then finally ligase will establish the phosphodiester bond between the vector and insert nucleotides, and that becomes the complete recombinant DNA molecule. Gap filling and ligation by host polymerases and ligases then you get a recombinant DNA molecule with the insert, right? You can see. So here we did not use the coincidence, but instead of that, insert and vector are homopolymerized, or homopolymeric tails are stitched at 3' end by using the terminal deoxynucleotide transferase, and then they are mixed and they're annealed. Annealed product is transformed into the E. coli. Then you see a recombinant DNA molecule can be purified from that E. coli. So simply, without, when a DNA fragments having no cohesive sites, then still you can clone by addition of the homopolymers at 3' end. In 1975, the first time this technique was used, so they used the they used the SV40 DNA and cloned and transformed into the mammalian cells. Later on, it became a part of the molecular cloning. So this is the last DNA joining molecule discussed in the early presentation, where we discussed about linkers and the adapters. So this is all about the homopolymer tailing of blunted DNA fragments by using the terminal deoxynucleotide transferase. Thank you all for listening the lecture. If at all you like it, try to subscribe my channel. That's Dr. K. PREM-PRIMER. And if you have any complaints or compliments, you can write to me through the comment section. If you like it, try to give thumbs up. If you want to share, you can share. Thank you all. See you again with one more video. Until then, bye-bye.